BBYCT 137, Plant Physiology and Metabolism, June 2023 paper. Question 1a. State whether the following statements are true or false. I. The water potential is less negative in the xylem than in the soil water. False. E. Both cyclic and non-cyclic electron transport operate in photosynthetic bacteria. False. E. In cyanobacteria, nitrate reductase is located in the photosynthetic membranes. False. IV. The sources and sinks for solute transport are always fixed. False. Answer 1a. 1. The first statement is false because the water potential in the xylem is more negative than in the soil water. 2. The second statement is also false since photosynthetic bacteria generally carry out only cyclic electron transport. 3. The third statement is false because in cyanobacteria, nitrate reductase is found in the cytoplasm, not in the photosynthetic membranes. 4. The fourth statement is false because the sources and sinks in plants are not fixed permanently. Their roles can change with the developmental stage or environmental conditions. Question 1b. I. The source of O liberated in photosynthesis is water HO. During the light reactions of photosynthesis, water molecules are split in a process called photolysis, releasing electrons, protons, and oxygen. E. The upward movement of water is known as the ascent of sap, which occurs through the xylem due to transpirational pull and root pressure. E. In the rainy season, the wood of doors gets swelled up due to imbibition, which is the absorption of water by hydrophilic substances like cellulose in wood, causing them to expand. IV. Magnesium Mg² and iron Fe², ions are required for chlorophyll synthesis. Magnesium is the central atom in the chlorophyll molecule, while iron is essential for the enzymes that facilitate its biosynthesis. Question 1c. Match the items in column A with column B. A. Molecular oxygen IV hydrogen acceptor. B. Electron acceptor E. Cytochrome C. C. Pyruvate dehydrogenase E. Acetyl-CoA. D. Decarboxylase I. Ketoglutarate. Question 2. Differentiate between the following pairs of terms. 2i. Photophosphorylation vs. Oxidative phosphorylation. Photophosphorylation is the light-driven synthesis of ATP that takes place in the chloroplast thylakoid membranes during photosynthesis. It can be cyclic or non-cyclic. In cyclic photophosphorylation, electrons cycle back to the same photosystem and only ATP is produced. In non-cyclic, electrons are transferred to NADP forming NADPH, and water is split to replace the lost electrons, producing oxygen. Oxidative phosphorylation, in contrast, occurs in mitochondria during cellular respiration. Here, electrons from NADH and FADH pass through the electron transport chain, releasing energy that pumps protons across the membrane. ATP is then synthesized as protons return via ATP synthase. The final electron acceptor is oxygen, forming water. 2e. Competitive vs. Non-competitive inhibition. In competitive inhibition, an inhibitor structurally similar to the substrate competes for the enzyme's active site. This can be overcome by increasing the substrate concentration. It raises Km but does not affect Vmax. In non-competitive inhibition, the inhibitor binds at a site other than the active site, changing the enzyme's shape and reducing its activity. This cannot be reversed by adding more substrate. Km remains unchanged, but Vmax is reduced. 2e. Diffusion vs. Osmosis. Diffusion is the passive movement of molecules from a region of higher concentration to lower concentration until equilibrium is reached. It doesn't require a membrane. Osmosis is a special case of diffusion where water moves across a semipermeable membrane from a region of low solute concentration to high solute concentration. Osmosis is vital for turgor pressure in plants and plays a major role in water uptake by roots. 2iv. C3 vs. C4 plants. C3 plants fix CO directly through the Calvin cycle forming a 3-carbon compound, 3-phosphoglycerate. Rubisco is the key enzyme, and photorespiration occurs under high oxygen and light. C4 plants first fix CO into a 4-carbon compound oxaloacetate using PEP carboxylase, which avoids photorespiration. CO is later released in bundle sheath cells for the Calvin cycle. C4 plants are more efficient under high light and temperature conditions. Question 3. Explain Krebs cycle. Highlight metabolic intermediates and sites for NADH, FOD, and ATP production. The Krebs cycle, also known as the citric acid cycle or TCA cycle, is a central metabolic pathway that occurs in the mitochondrial matrix. It begins when acetyl-CoA produced from pyruvate combines with oxaloacetate to form citrate. Citrate is isomerized into isocitrate, which is then oxidized to alpha-ketoglutarate, producing NADH and releasing CO. Alpha-ketoglutarate is further oxidized to succinyl-CoA, producing another NADH and CO. Succinyl-CoA is converted to succinate, generating ATP or GTP via substrate-level phosphorylation. Succinate is oxidized to fumarate, producing FOD. Fumarate is hydrated to malate, which is then oxidized to oxaloacetate, yielding a third NADH molecule. In total, for each acetyl-CoA, 3 NADH, 1 FOD, and 1 ATP are produced. These high-energy carriers are then used in oxidative phosphorylation to generate more ATP. The Krebs cycle is vital not only for energy production but also as a hub for biosynthetic precursors. Question 4. Define vernalization. What role does it play in agriculture? B. Explain active transport of metabolites through ionic pumps. 4a. Vernalization and its agricultural role. Vernalization is the process of inducing flowering in plants by exposing them to prolonged cold temperatures. It is essential for certain biennial and winter annual plants like wheat and cabbage. Cold treatment promotes floral development and prevents early vegetative growth from turning into flowering prematurely. In agriculture, vernalization ensures timely flowering and enhances yield predictability. Artificial vernalization techniques are used in breeding and controlled crop cycles. 4b. Active transport of metabolites via ionic pumps. Active transport involves the movement of molecules against a concentration gradient using energy, usually from ATP. Ionic pumps in plant cell membranes, such as proton pumps HATP-ases, generate electrochemical gradients by pumping protons out of the cell. These gradients drive secondary transport systems like symporters or antiporters. Nutrients like nitrates, phosphates, and sugars are transported into cells by coupling their movement with proton influx. This mechanism is crucial for nutrient uptake, phloem loading, and ion homeostasis. Question 5. A. Explain the mechanism of allosteric enzyme regulation. B. Discuss the mechanism of opening and closing of stomata. 5a. Allosteric enzyme regulation involves the modulation of enzyme activity by binding of molecules at specific sites called allosteric sites, distinct from the active site. When an effector binds to an allosteric site, it induces a conformational change in the enzyme that either increases activator or decreases inhibitor its activity. This regulation often occurs in enzymes that control key steps in metabolic pathways. A classic example is the enzyme phosphofructokinase in glycolysis, which is inhibited by ATP and activated by AMP, thus ensuring that glycolysis proceeds only when energy levels are low. Allosteric regulation enables the fine-tuning of metabolic flux and allows rapid responses to changes in the cell's internal environment. 5b. The opening and closing of stomata is a crucial physiological process regulated by guard cells. During daylight, light, especially blue light, activates proton pumps HATP-ases in the guard cell membrane, leading to efflux of H ions. This creates a negative potential, causing K ions to enter the cells. The accumulation of K and Cl ions lowers the water potential, and water enters the guard cells by osmosis, making them turgid and causing the stomata to open. In the dark or during stress like drought, abscisic acid (ABA) triggers the efflux of K and other ions, raising water potential and causing water to exit. This results in flaccid guard cells and closure of the stomata. The mechanism regulates gas exchange and water loss, balancing photosynthesis with water conservation. Question 6. A. Discuss the functions of essential elements. B. What is stress? Discuss temperature-induced stress. 6a. Functions of essential elements. Plants require 17 essential elements. for normal growth and metabolism. These are divided into macronutrients, e.g., nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, and micronutrients, e.g., iron, manganese, zinc, copper, molybdenum, boron, chlorine, nickel. Nitrogen is a key component of amino acids and nucleic acids. Phosphorus is part of ATP, DNA, and phospholipids. Potassium regulates osmotic balance and enzyme activation. Calcium stabilizes cell walls and membranes. Magnesium is the central atom in chlorophyll and activates many enzymes. Iron is crucial for electron transport and chlorophyll synthesis. These elements act as structural components, enzyme cofactors, and regulators of physiological functions like osmotic balance, signal transduction, and energy transfer. 6b. Stress and temperature-induced stress. Stress refers to any adverse condition that negatively impacts plant growth and metabolism. It can be biotic pathogens, herbivores, or abiotic temperature, drought, salinity. Temperature stress includes both heat and cold stress. High temperatures can denature proteins, destabilize membranes, reduce photosynthesis, and increase respiration, leading to cell damage or death. Cold stress affects membrane fluidity, slows enzymatic reactions, and may cause ice formation, damaging tissues. Plants cope by producing heat shock proteins, altering lipid composition, and synthesizing osmoprotectants like proline. Stress-tolerant varieties are bred to ensure productivity under extreme conditions, making temperature stress management crucial in agriculture. Question 7. Write short notes on any two of the following. 7i. Protosmotic model for phloem transport. The protosmotic or pressure flow model describes the movement of sugars in phloem as a bulk flow driven by pressure differences. At the source, like leaves, sucrose is actively loaded into sieve tubes, reducing water potential. Water follows by osmosis from adjacent xylem vessels, creating high turgor pressure. At the sink, like roots or fruits, sucrose is unloaded, increasing water potential, causing water to leave and lowering pressure. This pressure difference drives sap from source to sink. The model explains rapid and bidirectional transport in phloem. 7e. Photorespiration. Photorespiration is a process that competes with photosynthesis in C3 plants. When rubisco binds O instead of CO, it produces phosphoglycolate, a 2-carbon molecule that cannot enter the Calvin cycle. The plant recovers some carbon through a series of reactions in peroxisomes and mitochondria but loses energy and releases CO. It is more pronounced under high temperatures and light. Photorespiration reduces photosynthetic efficiency but may protect plants from photooxidative damage. 7e. Bioassays for auxins. Bioassays are experimental methods used to measure the presence and activity of hormones like auxins. Common auxin bioassays include the Haute coleoptile curvature test, root elongation assay, and hypocodal swelling test. In these tests, plant tissues show measurable responses such as bending or elongation in reaction to different concentrations of auxin. These assays help in detecting natural auxins and comparing the effects of synthetic analogs. 7iv. Phytochrome-mediated responses. Phytochromes are photoreceptors that regulate plant responses to red and far-red light. They exist in two forms, PR inactive and PFR active. Exposure to red light converts PR to PFR, while far-red light reverses it. These changes regulate seed germination, shade avoidance, and flowering. Phytochrome signaling integrates light quality with developmental processes, enabling plants to adapt to their light environment.